what does it say? It says there'll be a slight delay. And then that little red spot will go to green. Why is it not doing that? Oh, it says it's doing it, but that's still red. Once a recording is started, the icon will change to indicate a recording is taking place. Yes. And the light on the desk... Oh, okay, sorry. That's supposed to go green. Okay, right. It is recording. Right. But it's not full screen, is it? Which is peculiar. Can you see that all right, though?
Just click F5 on the keyboard.
F5. Cancel. There we are. Very good. Right. But you know who I am. So, reporter gene technology. Basically, it's when you link a well-characterized gene or gene product to a gene or gene locus to allow a technology that's already well established and easy to investigate something that could be quite difficult to look at. So looking at gene expression, gene regulation, or the location of a protein or set of proteins within the cell. And so you can think of it as being a fusion that allows you to look at transcription, looking at translation, or looking at location. Okay? So the reporter is that well-established... Can you take a copy of the handout, please? The well-established gene or gene locus that you're going to use. Okay, so I am a microbiologist, a bacteriologist, so while a lot of the principles that I'm going to be talking about apply to both prokaryotes and eukaryotes, I will be unashamedly concentrating on bacteria. Okay? There are a couple of examples that I put in about eukaryotes. One of the problems, it's much more difficult to establish your fusions in a eukaryotic system than it is in a bacterial system, because for a variety of reasons. So often the constructs in a eukaryote are made and you get transient rather than stable expression of the reporter system. So one of the things that you need to think about with reporter genes is how you actually detect the activity of the reporter that you're looking at. And so it could be cytochemical, which is when you add a chemical to a set of cells and that chemical gets taken up and then gets converted into something that can be detected actually in situ in the system. So looking at, if you've got a set of cells, you can tell which cells are actually active for the particular activity you're looking at. Or it could be that you're actually doing it on, by colony. You're looking at which colonies are made up of bacteria expressing the reporter gene. Okay? And so a good example of that is the blue-white color difference that you get with a lot of gene cloning vectors when you use XGAL as the chromogenic substrate. that same sort of system of detecting activity may well allow you then to also do a biochemical assay to actually get quantitative data on the amount of activity. And so you might produce extracts of cells and use a substrate like orthonitrophenyl galactoside, which is the substrate for beta-galactosidase that goes from colorless to yellow, and that transition from producing yellow to produce yellow color can be quantified either by taking a determination at an endpoint or actually following yellow color production in time in a spectrophotometer. It might be that you actually use fluorescence, and so GFP, green fluorescent protein, is an example of that. Obviously there it's a question of exciting the reporter with an appropriate wavelength and measuring the output, the fluorescence, at a different wavelength. Can you pick up a copy of the handout, please? And then finally, light emission is a really good example where as long as you're providing a substrate, if it's not produced endogenously, the reporter will allow light emission, again proportional to the amount of reporter gene activity. So the key point from this is always with the reporter, think about how do you detect and what limitations does that put on how you can use it. Because the more complex the system that you're putting your reporter into, the more complex, the more limitations there may well be on which reporter you use. Okay. And so some examples of reporter genes, beta-galactosidase, well, the LAC-Z gene encoding beta-galactosidase was probably the first reporter gene, partly because it was so well known, it was the first gene to be sequenced, and so knowing stuff about LAC-Z, that the chromogenic substrate XGAL, the standard assay uses ONPG, and I put IPTG there. Who knows what IPTG does? Someone say it out loud. What? It induces expression of the LAC operon. Yeah? Okay. So it's not directly applicable in all the cases where you use LAC-Z as a reporter system, but it's just something, the LAC operon is a system that you should know as a paradigm because it comes up so many times in all sorts of contexts. Okay. So the second one I've put here is CAT. So that's chloramphenyl acetyltransferase. And you can assay that. There's a chromogenic assay for that. But I've put that there for to get you to think about, anyone have an idea of how else you could detect CAT activity? Other than a biochemical assay. Okay, I'll ask you in a different way, different question. What is chloramphenicol? Yeah. Okay. So, what do you think CAT does?
We can screen it. If a plasmid has the CAT gene, we can screen it on other plates.
How?
In different dilutions, if you have chloramphenicol, and if you plate it, and then you observe the zone of inhibition.
Yeah. Okay. So, CAT that activity, that biochemical activity, chloramphenicol acetyltransferase, it transfers an acetate group onto chloramphenicol, and in doing that, it inactivates the antibiotic. Okay? So it confers resistance. So actually, the phenotype that you can get from CAP expression is resistance to chloramphenicol. So, and the more expression you get, the more enzyme you get, the higher the level of resistance. Okay? Does that make sense? So if you want to know whether you've got a promoter that gives any expression at all, you could link the CAP gene to it and ask, do you get chloramphenicol resistance? You can actually select for those constructs that actually put a promoter upstream of the CAP open reading frame. Okay? So I'm sort of emphasizing that just to get you to think about, it's not rocket science, it's just tinkering. It's sort of like synthetic biology. You're actually using genes, the bits of genes, to tell you about other bits. Okay? So you're using the open reading frame. Hello, do you want to pick up a copy of the handout? And can I emphasize that those people who came in late, there is a sign-in sheet that you need to sign, and it was going around somewhere. Okay? Right. So then these are just other examples. Xylee, which is, well, encodes catechol 2,3 oxygenase. You'll find that used in a variety of, particularly outside of E. coli. GFP, we'll think about that a lot more in a little while. GUS, encoding glucosidase, it has been used in plant systems quite a lot, and LUX, luciferase that gives light emission. Okay, so, and if associated with this module, you're going to write an essay about a paper, what you want to do is to think about, the particular, the reporter that's been chosen, why it's appropriate for that particular application. Okay. Okay, so I think the first sorts of reporter assays that people started doing a long time ago was actually looking at measuring transcription through a reporter gene. And so the couple of problems I've got for you later on is very much based on thinking about having a reporter gene in a plasmid where we've got a replication system, a selectable marker, a reporter gene, and a multiple cloning site. And the multiple cloning site allows you to insert DNA fragments that might have a promoter on them and direct the expression of this gene. So sort of by definition, this reporter gene will have the open reading frame, the coding region. Now if you look down here, it shows in slightly more detail, you'd have the open reading frame preceded by a ribosome binding site, so that if you get messenger RNA, you will get translation of that open reading frame. And then here we've got the restriction endonuclease site of the multiple cloning site where you can put your fragments. But normally one of the things that would be in a generalized design would be to have stop codons here so that, can you pick up a copy of the handout, please? You would have stop codons in all three open reading frames so that a fragment inserted into this location, if it contained the beginning of an operon, for example, it would have a promoter, and then it would have the transcription start, and then it would have translational signals, and you'd have the first open reading frame. In order to simply get something which is an uncomplicated measure of the amount of transcription that's coming through, you would want any open reading frame in the fragment you've got inserted here to stop before you get into your reporter gene. Okay? And so nowadays, with synthetic DNA being so easily available and working off DNA sequences, you would generally not use a sort of off-the-shelf reporter system like this. You'd probably design the fragment to suit the particular vector you're going to use. But the same principles apply. You need to have a place where you're inserting the DNA to be tested. You need to make sure that it is not sending signals down into the reporter system that are going to screw up the expression of the reporter gene. And so you end up with this sort of generalized design. Reporter gene. And so the point about this is that you do not need the inserted DNA to create a translational signal. The translational signal already is there. You're simply wanting to find out whether you've got a promoter in the fragment that's inserted there. Okay? Is everyone happy with that? Okay, so one of the problems with a high copy number plasmid, or even a moderate copy number plasmid, is that you will have many copies of the gene. And so if the normal situation is that you have a single copy of the gene in the chromosome, actually raising the number of copies in the cell may alter the way in which a promoter region behaves, particularly in its regulation. Okay? So I want you to think about that for a moment. How do you think changing a copy number could alter the behavior of a promoter and regulatory region? Well, it is amplified by putting it onto a plasmid.
But even more so.
Why? I mean, I'm not saying no, it's not, but I mean, but I was wanting you to think about the problems of just the amplification that results from it being put on a high copy number plasmid. It could do. It could do. So you could get that negative effects of having multiple copies or large amounts of expression from the gene that you're looking at. That you could do, yeah. Any other? But I want you to focus actually on, what you're inserting here. Think about let's go back to the paradigm of the lac operon. How many of you could tell me something about the lac operon as a system? Put your hands up. Not many people admitting to it. Okay, but so what do you know about the regulation of the lac operon? I wouldn't say it's a negative. It is negative, but it's not negative feedback. The lac operon is repressed by a lac repressor. Does that ring a bell? Okay. And the lac repressor is encoded on the chromosome a little way away. And actually, the number of copies of the lac repressor that lac I gene produces is very small. But because of the affinity of the repressor for the operator that allows it to repress, near the promoter, those small number of repressor proteins are actually normally located on the sitting on the operator. Okay. What would happen, do you think, though, if you put that promoter operator region on a multi-copy plasmid? Come on, you're supposed to be thinking. What would happen? You've got a small number of repressor proteins floating around the cell. You've got one, normally you've got one promoter operator region in the chromosome. Sort of. So you would actually use up all the repressor, and there might then be promoters for which there aren't repressor proteins. Okay? So by putting your your promoter region onto the multi-copy plasmid, you could end up destroying the repression because there isn't enough repressor to repress all those promoters you've now got in the cell. Does that make sense? You're titrating. It's not saturating. You're titrating out the repressor. Okay? So, depending on how you use your reporter, you might well end up screwing up the regulatory system that you're trying to investigate. Okay? And so an alternative, other than putting, alternative to putting your promoter region into a reporter vector, you can put your reporter gene into the gene in the chromosome that you're wanting to investigate. Okay? Does that make sense? So this is just showing you inserting the reporter system into the gene to be studied and recombining that into the chromosome so that now, instead of putting your promoter into the vector, you're putting the reporter into the gene. So those are two different ways of constructing your transcriptional fusion. And in this situation, here we've got our open reading frame, and that's why it's really important that we have our stop codons here so that they will cause this open reading frame to terminate, and then you'll have like another gene. Now your reporter gene becomes effectively the second open reading frame. Well, the next open reading frame after the beginning of this gene. So I'm just trying to get you to think about, the way in which you would put your reporter gene and relative to the expression signals you're trying to study. Is that making sense? Yeah? Okay. So that's just a summary. The reporter gene, if it's a transcriptional reporter that you're looking at, it has its own ribosome binding site. It's preceded by stop codons. It's important to try to get the fusion to the same copy number as the original gene. And the other thing that I haven't said there yet is that if you do this, you've got to think about what putting that gene in there might do to any downstream genes. Okay? Because someone said negative feedback. It might be that the system you're looking at is regulated by a gene in this operon. If there is a series of genes, and if, say, a regulator for the promoter up here is down there, and your reporter goes in and then blocks the expression of the downstream genes, you might again screw up the regulatory system. Does that make sense? So really, reporter genes are sort of simple in principle, but you've got to be thinking very, very carefully about how you use them. Okay? Because you want to investigate a real system, you don't want to investigate an artifact of actually creating the fusion to your reporter system. Okay? And so this just illustrates an additional sort of vector. Some promoter regions are bidirectional. They've got promoters firing both directions. And so if you want to measure two directions at once, you can have a reporter system which has genes firing away from the cloning site, different genes, so that you've got two different ways of measuring the transcription from the two directions. It just allows you a bit more flexibility. Okay? But otherwise, the same principles apply. And if you're dissecting a regulatory system, you might want to not just look for where a promoter is, you might want to look for the regulator as well. And that's much easier. If you're studying E. coli, for example, it's slightly complicated because you obviously might be trying to work in E. coli, pulling out promoters and regulators, and they're already there in the chromosome. Okay? Whereas if you're using E. coli as your system for studying the gene expression, if you take genes from quite a different organism, then they're not going to be so the chromosome effectively is just the way of the bug growing, and your promoter might come from a totally different bug and wouldn't have its cognate regulator in E. coli. And then you could clone genes either based on, you've got a hunch that this gene might be involved, or you can make a library of genes to find a gene that regulates the promoter that you've captured. But basically what I'm trying to get you to think of is that if you've made a reporter gene fusion to a promoter, you can then use the activity of that reporter gene to look for genes that either upregulate or downregulate that reporter gene. Okay?
Would the bidirectional
Could be.
if they're functions
Yeah, absolutely. I mean, there are switches which actually are bidirectional, but one direction is on or the other direction on. It's a sort of flip-flop, and you could detect that sort of change if you could detect both of those reporter genes effectively at the same time or on different sorts of plates. Yeah. And it's in this situation where, if you're looking for a regulator, you might well put the regulator under the control of a regulated promoter, so you could switch the regulator on or off, and that might mean that you were using a common control switch in E. coli. A lot of the control switches in vectors in E. coli are regulated by the lac repressor, and therefore, switching this regulator on or off by adding IPTG would be something you might build into an experiment. And so a typical experiment would be having your reporter gene alone, because quite often the reporter, the reporter vector alone, without the inserted fragment, quite often you get a little bit of background of activity. Okay? So you'd need to know what the background is. You have your vector with the promoter inserted into it. And then here you'd have the second plasmid. So these vectors would be different. This would be the reporter vector. This would be the expression vector. You'd have your empty expression vector, and then the vector with the putative regulator in it. You'd add the inducer, plus and minus the inducer, and you'd end up looking at the effect of this regulator on the activity of the promoter. Does that make sense as a sort of experimental design? And so a typical experiment would be you'd have overnight cultures of bacteria, you'd dilute them and grow them into log. It says lag phase. It should be log phase, okay? So exponential phase. I hadn't noticed that before. Lag phase is when you get to the stationary phase. So you'd add the inducer and grow for a bit further, harvest the bacteria, or actually just measure activity as the culture grows, and then you would determine the amount of reporter gene activity you have. Okay, so these are caveats that I've already mentioned. You need to make sure that the fusion gives a fair reflection of normal gene activity. One of the things that you need to worry about is that messenger RNA, if you're interested in expression, messenger RNA is actually a regulatory element within systems. So messenger RNA for most bacterial genes is quite short-lived. It'll degrade very rapidly. And so by fusing a promoter to an unnatural gene, your reporter gene, you might actually be altering a key property such as messenger RNA stability. And the other thing is that if you're looking at the effect of a repressor or an activator, you need to think about how long does it take the growth of the culture after you've switched on the regulator to get a steady state. And so, if you're in that simple experiment that I was talking about, the bacterial culture will be growing. You add the thing that'll switch on the regulator, and it'll be a while before you start to see the big difference between having the regulator there and having it not, depending on whether it's an activator or a repressor. Okay, so again, using a reporter, you've got to do it with a knowledge that, depending on how you do your experiment, you could end up with really very different results. Okay, so experimental design and care and controls are really important. So this is just an example of the sort of thing that people have done in eukaryotic systems. Eukaryotic promoters are generally much more complicated than bacterial promoters, and generally they're much more dependent on activators than repressors. Okay, so in a typical investigation, you might well be looking. You've got your reporter there, and a typical experiment would actually be to create a series of fragments, effectively making shorter and shorter fragments, actually asking what do you need for promoter activity and what changes the activity in response to different factors that affect gene expression. And these deletions would define regions where activators bind and are needed to switch on promoter activity. So you make those fusions and then transfect and simply look at gene expression from the reporter under different sorts of conditions, for example, stress conditions or oxygen level or heavy metals, all sorts of different, or hormone levels. Okay? Yep.
How long are promoters using this generally?
In bacteria, the key elements are focused at -10 back from the transcription start point and -35. So within 50 bases, you would have the important parts for RNA polymerase binding. But activator binding sites are generally located further upstream from that. And so you probably need 100 bases to be confident that you've got the most obvious transcriptional signals. Okay? If you're talking about a repressible system, you would normally need only about 40 bases, because normally repressor binding sites overlap either the -10 or the -35 region. But having said that, one of the interesting things with many repressors is that actually looping is important for repression. And so the LAC repressor, well, no, not the LAC. Yeah, sorry. The LAC repressor is not such a great example, but there are quite a few where a second binding site for the repressor can form a sort of, the two binding sites can form a loop which actually shuts down the promoter much more effectively. So it's quite difficult to answer your question very precisely, but does that help?
Yeah, I was just wondering how long it would take to sort of like study the...
Well, in a eukaryotic system, it can be kilobases. And so eukaryotic promoters are generally much more complicated. They do have equivalents of those, well, certainly of the -10 region. The tartar box in the eukaryote is critical, but then these activator binding sites can be kilobases away. So, and chromatin is so much more complicated. I'm certainly not an expert on those. Yeah. Okay. So reporters aren't just used for academic purposes. Some are actually used as biosensors. So bacteria have been exploited, for example, Lux as a reporter system linked to the Xyle promoter. So there are soil bacteria, Pseudomonas putida, that carry genes that allow them to degrade things like xylene, toluene, organic solvents. And often on industrial sites, you've got organic solvents that have sunk into the ground and need to be remediated. And so they need to know how much solvent there is there, or which soil samples have those solvents present. And so they've made bacteria. So the Xyle the Xyle genes are those degradative genes, and they're switched on by the presence of organic solvents. And so their promoters respond through regulators, Xyle S and Xyle R, to the presence of solvents. So if you make a reporter and you put the bacteria there, if the light comes on because this promoter has been activated, it tells you that there's organic solvent in that sample. And then this second example, the LAS promoter regulates elastase in Pseudomonas aeruginosa, and so that is under control of what is called quorum regulation. So during infection, the bacteria start to grow, and then they get up to a certain density, and then they start to produce degradative enzymes that allow them to spread. And that density is sensed by homoserine lactones. And so if you want to know whether there are those homoserine lactones in a bit of tissue that seems to be inflamed because of infection, you can use this reporter system, and it'll tell you whether those signals are present. Okay? So there are lots of ways in which you can use reporters in a practical sense. And many of these sort of applications are patented. Okay. Right. So that was just getting you to think about transcriptional fusions. Translational fusions are very, very similar, except that instead of having a ribosome binding site and a start codon at the beginning of the gene, you just take the open reading frame minus its translational signals. So you don't have a ribosome binding site, you don't have a start codon. You rely on fusion to the system that you're investigating to create those signals. Okay? So here we've got the open reading frame, and here you've got places where you can insert the system to be investigated. And if you create an open reading frame, so if you have a start codon and a series of triplets that then link into the open reading frame for your reporter, that allows you to detect how much transcription and translation is coming from the bit of DNA to be investigated. Okay? And if you do two sorts of fusion, a transcriptional fusion and a translational fusion, and compare the results from those, you can actually see at what level regulation is occurring. Okay, because obviously you can control expression through both levels. You might have loads of transcription, but actually very, very poor translation. Okay? And I can talk to you for a long time about the sorts of signals you might see, but the point is, I'm just wanting you to think how you could use that principle of reporter. Okay? This is an example of using the LAC-Z gene in looking at regulation in eukaryotes. It's really quite an early use of that, and looking at developmentally regulated gene expression in Drosophila, and so making actually integrating LAC-Z into the chromosome downstream of various promoters which were thought to be developmentally regulated. And so in the Drosophila larva, you get these segments, and what you can do with a chromogenic substrate is simply dunk the larva in a solution. It's sufficiently transparent and sufficiently easy for the chemical to diffuse in. When it gets into the cells where the enzyme is being expressed, you get the change to the blue color, and so you can see the precipitation in those cells within the complex eukaryotic system where those genes are being switched on. So that's actually both a transcriptional fusion and a locational fusion, because it's telling you where in the organism expression is being activated. Okay. And so that then leads us on to that third sort of application, and that is locational fusions. And as far as bacteria are concerned, that has been revolutionary in terms of being able to see where inside bacteria things are happening. And so we've used them in plasmid, looking at plasmid partitioning. And so this shows a fusion between GFP and an ATPase, which forms filaments and which is sort of like the mitotic apparatus in eukaryotic systems. And so what is illustrated here is that if we have GFP on its own being expressed, it diffuses throughout the bacterial cell. If we have the fusion between INC and GFP, it diffuses throughout the cell. But if we then put a plasmid in that normally uses this protein and a DNA binding protein, which we call CoRB, for positioning the plasmid DNA in the cell, then actually the picture changes, and now all of the GFP, you get a peak of GFP forming a focus at one point in the cell. And we now know from a lot of experiments that what happens is that the plasmid DNA, these proteins plus the plasmid DNA cause foci to form. That is, the plasmids clump together, and that involves CoRB protein binding to the plasmid DNA, and when that is bound to the plasmid DNA, then the ATPase activity protein binds to that, and it forms a complex. And you get these foci, but then when at appropriate stages in the cell cycle, the foci split and move away from the division plane. But this, the movement of the fusion to a particular part of the cell is telling you something about the function of the proteins to which you've fused. And so this just shows a sort of time lapse for a cell that is growing. You can see two of these foci, and as time goes on, then this one splits and you get two. That one is splitting and you get two. And eventually, as the cells grow and grow, you have four, and then the cell will divide, and then these will move to the opposite poles. Okay? So that's telling you where the plasmid and its associated proteins are located in the cell. Okay? But again, if you look at the literature about this sort of study using GFP, you will discover that in the early days, a lot of the experiments were totally artifactual. People were using reporters and producing nice results, which turn out to be an artifact of the way in which they were actually doing the experiments. Okay? So you've really got to take reporter gene stuff, and particularly this sort of subcellular visualization of things in complex systems, with a real pinch of salt. And one of the things you should do if you're writing an essay on this area for this module is be skeptical, okay? Don't take conclusions at their face value. Always ask, what's the basis of that conclusion, and is it valid, okay? Seeing is believing, but believing is a bit naive unless you really question what it's telling you. Okay? And I'll leave those examples. Those are just other examples. Okay? So on the theme that I was saying, one of the things that you need to do is to make sure that any fusion between a reporter and a protein does not screw up the normal function of the protein that you've made your fusion to. So, question. How would you tell that the normal function has been retained?
Your phenotypes.
Yeah? Okay. Go on.
So you could look at things like growth or reduction, like you could assay kind of what's produced by the cell.
Yeah. But tell me a little bit more about what would you do?
I need to think of a protein.
Okay. But in general, I mean,
I think you just compare them. Just
what do you mean compare what?
well, you'd compare your regular normal protein with no reporter, and then you compare that with your protein with a reporter, and just see whether the results are different.
Okay, but you're missing out what I think is one important thing about how that is set up.
Oh, you still need to attach something to the protein, do you?
No, that's not what I'm thinking about. What am I thinking about? Someone else? Or, I mean, if you want to know whether your fusion is functional, in what cell will you test it? But will you test it?
Same type of cells that you're going to test your normal.
Well, not exactly. What's different about the cell that you're going to test it in?
Eukaryotic bacteria.
Doesn't matter. Absolutely. Yeah. If you have a deletion of the normal, if you can do that, and the question is, can you complement that mutant with your fusion? And does it make it normal? So you're absolutely right, but you just missed out that. Do you see what I mean?
Yeah.
Because if it was the normal protein that was keeping it normal, you need to get rid of that. Okay? Okay, so with that in mind, at some stage this green light is going to go red. But anyway, you need to think, how do I make sure I've got a functional fusion? So what this illustrates is, first of all, you can make N-terminal fusions. You can get C-terminal fusions. Okay? There's nothing magic about which end you make your fusion to. So you can do it both ways. And you can put a linker in. So the key thing is that you're making sure that what you've got, the two things are tethered, but you need to end up with a functional protein. Okay? Is that all right? The next thing is that GFP can be quite stable. So if you're looking at expression or changes in expression, once you've switched on GFP, it hangs around. Okay? And so if you were looking at a system where, what you're reporting on is getting switched on and then switched off, you might not see the switch off because the protein just hangs around. Okay? And so one thing that people have done is actually to tag the reporter with degradation signals, so that the reporter is actually relatively short-lived, so that when it gets switched on, you see a signal. When you get it switched off, the signal disappears. That means that the whole system, actually you never build up such a high level of the reporter, and it's more difficult to detect the reporter. But it means that, and so you need a sensitive system for detecting the reporter, but it means it is more realistic. Okay? And so this is yet again about the sort of worrying about what the results that you see mean. And so continuing on this theme, one of the really neat applications of reporters, locational reporters, allows you to ask where in a cell is a gene located or a part of a chromosome located. And so using the lac operator, going back to the lac operon, this is why I was emphasizing at the beginning the paradigm of the lac operon. So the lac repressor binds to the lac operator. We know a lot about the lac repressor. We know a lot about the lac operator. You can make cassettes which have multiple copies of the lac operator. Okay. And the original ones, because the microscopy was quite insensitive, used cassettes of something like 256 operators in a row. And then they made fusions between the lac repressor and GFP, so that when that was expressed in the cell, it would go and bind to the cassette of those operator sequences. Does that make sense? And so if you insert your cassette into a part of the chromosome like the telomeres, or close to the telomeres, or near the centromere, you can actually see where they are in the cell because of where GFP goes when it's bound to your cassette. Okay? So that is a locational fusion. And now with increasingly bright fluorescent proteins and increasingly sophisticated microscopy, you can get away with these cassettes being much smaller and being, I think we can now do, if you've got a very good microscope, a single binding site for a single fusion between LAC-I and GFP. But, you know, so if you think about the size of a cassette of 256 copies of the operon, it's actually quite a long bit of DNA. And so you could be worried, well, heck, that can stretch across a bacterial cell. How do I know that I'm going to be able to see a very precise location? So the smaller the binding site region, the more accurately you can actually tell where your probe is. Does that make sense? And can you start, maybe you were quite familiar with this already, but if you weren't, can you start to see that there are all sorts of questions you can ask using reporter systems, and really the question is only limited by your ingenuity in what you think, how you think the question, what the question is going to be, okay? So people have tagged the replication origin in E. coli and just watched what happens to the replication origin in the bacterial cell as you go through growth and division. During sporulation, people have looked at which genes are expressed in the pre-spore compartment versus the mother cell compartment. Okay. And this is a really neat example. So we're looking at plasmid transfer, okay? So this is set up so that in plasmid transfer, we've got bacteria that don't have the plasmid, and we've got bacteria that do have a plasmid.